Hello everyone, welcome back to my channel. I hope you guys are doing well. And today we will be continuing the chapter which was pending. All right. So let's continue from the topic which we just left in the last session. So in the last class, what we did was we were learning about the topic, the growth rate, isn't it? There were two types of growth rate. One was arithmetic and the other one was the geometric. And we saw how the division is happening, how in the arithmetic and how the geometric growth rate differs from each other. All right. And we need to understand how the plant growth is determined through these. And we have also seen that the geometric as well as arithmetic growth or growth phase is seen during the embryonic development. All right. So we can see both of them. However, they are quite different from one another. Then we also discussed in the last session about the phases of growth, how the different phases of growth occurs in the plants starting from the growth rate, which is meristematic phase of growth, then comes the elongation and finally maturation. So how these changes are happening over the period of time, that we have learned in the last session. Now let's understand the topic today. Let's start from conditions for growth. So now when we talk about growth in plants, you must have gone ahead and done an experiment on germination of seed. So what do we see during germination of seed? Do you think a seed is going to germinate if you do not provide them with optimum condition? All right. So what is an optimum condition? It is the necessary condition required for the seed to grow or required for the plants to grow. Now, what are the things which are required by the plants? The plants may require water, oxygen, nutrients. All right. These are very, very useful and essential elements for the growth, you can say. Now, when we talk about plant cell, they grow in size, okay? They will grow in size by cell enlargement. So this was the initial cell, okay? And this cell has undergone the enlargement. Now, how is the cell enlargement happening? All right. How will the cell enlargement happen? The cell enlargement is going to happen with the use of water. This is very, very important. Once the water enters the cell is going to enlarge. All right. So plant cell grow in size by cell enlargement. This is very, very important, which in turn requires water. So water is an essential element for the cell to transform into an enlarged cell. So what happens is the turgidity of the cell helps in extension growth. Okay. So what happens is, what do you mean by turgid? Turgidity is nothing but the hardness, or you can say the stiffness which is being provided, and it is going to help in extensional growth. Okay. It helps in extension, or you can say it will help in growth. Thus, plant growth and further development is intimately linked to water status of the plant. So what do we see is water will lead to cell enlargement. All right. Cell enlargement will happen through water, with the help of water, because water is going to make the cell turgid, which will help in extensional growth, and further the cell is going to undergo enlargement. Okay. So now water also provides a medium for, apart from the plant growth. Thus, plant growth and further development is intimately linked to water, that we already discussed. Now, what happens is water also provide the medium for enzymatic activity. So, a lot of enzymatic activity is provided. Not only it helps in cell enlargement, it also helps in enzymatic activity. You know that there are various water-mediated enzymatic reactions. All right. So water also provides a medium for enzymatic activities needed for growth. Oxygen helps in releasing metabolic energy. Okay. So oxygen is also used in releasing metabolic energy, which you can see during photosynthesis as well, how oxygen is utilized. How the CO2 will be utilized and how the oxygen is going to get liberated. Okay, so oxygen helps in releasing metabolic energy essential for the growth activity. Nutrients. Now, oxygen, you have seen that how oxygen is useful. Now, nutrients. Now, what kind of nutrients will be required? So, let me take a different color pen over here. So, you can just understand macro and micronutrients which are necessary and are required by plants for the synthesis of protoplasm and act as a source of energy. So, nutrients are working in two ways. One, it is providing the energy. Okay, it required by the plant for the synthesis of protoplasm. You know what is protoplasm? Cytoplasm plus nucleus. That is your protoplasm of a cell or living content of the cell. You can also say it as living content of cell. That is basically your protoplast and act as a source of energy. So now we know what are the functions of water. We know the functions of oxygen as well as the functions of nutrients. Now let's understand, in addition, what are the other things? Very importantly, temperature. Okay, so every plant requires an optimum temperature range best suited for its growth. Okay, optimum temperature is required. Any deviation from these range could be detrimental to its survival. What do you mean by detrimental? It could be dangerous, or you can say damaging as well. Okay, it is going to damage its survival, or it is going to hamper, okay, of its survival. Environmental signals such as light, gravity also affect certain phase or stage of growth. So we have also seen that the other things are also very, very important, such as light, gravity also has an impact on its growth. So I hope so this paragraph is absolutely clear, which talks about how are the plant's growth dependent on certain factors like water, oxygen, and nutrients. Let's proceed to the other part now. Now, when we talked about macronutrients, macro is which is needed in large amount. Okay, need in large quantity. Okay, that is your macronutrients. Okay, all right. Now, macronutrients, as the name suggests, nutrient required by the plant in large amount. This includes, what are the macronutrients? Carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, calcium, and potassium. All right. And carbon, hydrogen, and oxygens are obtained from air and water, while the others are obtained from soil. Okay. And micro, the name only suggests that which is required in very less quantity, are the ones required in smaller quantities by the plant, which includes zinc, boron, etc. In addition, the other elements could be silicon, cobalt, selenium, sodium, which are present, which are required in higher plants. I hope this paragraph is absolutely clear. Conditions for growth. Now we need to understand few important things, which is very, very important over here, which is the differentiation and re-differentiation. All right. This is very, very important. What is differentiation? Dedifferentiation and re-differentiation. Very, very important. Now we should know that we already know that when we talk about a plant has the shoot apical meristem, which we call it as SAM, or root apical meristem, which is RAM. All right. Now, when the plant grows, we see that during the growth phase, meristematic tissue plays an important role. Okay. So when the cell divides, okay, or the cell derived from root apical and shoot apical meristem and cambium differentiate and mature to perform specific function. We know that how these cells are going to divide further and going to undergo a specific function, and they will undergo and differentiate to form a specific structure, which is going to form specific function. So this act leads to maturation, that is known as differentiation. Very, very important. What is differentiation? And all these terms can be understood with this short diagram. So you can see that this shoot apical meristem, root apical meristem, they are undergoing cell division because they are meristematic tissue and they have a lot of potential to divide continuously. Now, what is going to happen is when the meristematic tissue divides, this is the first meristematic tissue, it is dividing and forming two daughter cells. One is participating in the other works and the other one is going to undergo differentiation. Okay, this undergoes Further differentiation to form permanent tissue. Okay. When the meristematic tissue forms permanent tissue, or when the cell takes up permanent shape, size, and function, we call it as differentiation, which you have studied in grade 9. Now, this meristematic tissue is going to form a permanent tissue by the process known as differentiation. So, what is differentiation? Basically, it is the process of taking up of permanent shape, size, and function. This is known as the differentiation. Now, this permanent tissue, which is formed from the meristematic tissue by the process of differentiation, no longer divides. It's not going to divide further. So it has lost their ability to divide. And once this permanent tissue regains the ability to divide and forms meristematic tissue back again, we call the process as dedifferentiation. Okay. So what happened is initially meristematic tissue transformed into permanent tissue by the process of differentiation, right? Now this permanent tissue will again regain the property of dividing further and form meristematic tissue, and we call it as dedifferentiation. All right. Now again we have got a meristematic tissue. This meristematic tissue is again going to form a permanent tissue. All right. When it has undergone permanent tissue formation by the process known as re-differentiation. So, how we see that the meristematic tissue is getting transformed into permanent tissue, and further permanent tissue is getting transformed into meristematic tissue, and this basically redefines differentiation, dedifferentiation, and re-differentiation. I hope this is absolutely clear. Okay, so let's understand what the NCERT says. So this act leads to maturation and is known as differentiation. So what happens is during differentiation, let me tell you some important points over here. So what happens is during differentiation, cell undergoes few major structural changes, both in their cell wall and protoplasm. Now this transformation from meristematic tissue to the permanent tissue will not happen just like that. There will be few changes. What are those changes? Basically, structural changes. What are the structural changes happening? Where is the structural changes happening? It is happening in the cell wall and protoplasm. For example, to form a tracheary elements, you know what is tracheary elements? Xylem and phloem. The cell would lose their protoplasm. So in order to form a tracheary elements, for the plants to form tracheary elements, they are going to lose protoplasm. They also develop very strong elastic lignocellulosic secondary cell wall. Okay. So they also will try to develop this cell wall, which is made up of lignin and cellulose. This is very, very important to carry water to long distance, even under extreme tension. Okay. So these are the features which have been observed when we see that how the meristematic tissue is transforming into a permanent tissue. So what are the changes do we see? Loss of protoplast to form tracheary elements, and they also form a strong elastic lignocellulosic secondary cell wall because it has to carry water to the longer distance under extreme tension. Okay. Now, plant shows different interesting phenomena. What are these? The living differentiated cells that by now have lost the capacity to divide can regain the capacity to division under certain condition, and we call it as dedifferentiation. Just now I told you what is dedifferentiation, when the permanent tissue is able to transform into meristematic tissue. Now the permanent tissue will regain its ability to divide and it is going to form meristematic tissue, and we call it as dedifferentiation. Ability to regain the capacity, this is very, very important or under certain conditions. Okay. For example, let's take an example over here. What does the example says? Formation of meristem, intrafascicular cambium and cork cambium. Okay. When we see that formation of meristem, okay, interfascicular cambium or cork cambium from fully differentiated parenchyma cell. So you know that parenchyma cell is a permanent tissue. It's a permanent tissue. Okay. So this permanent tissue, these are simple permanent tissue. Okay. So these permanent tissues, when they are going to get transformed into intravesicular cambium or cord cambium from fully differentiated parenchyma cells. Okay. So this is one of the example of dedifferentiation. While doing so, such meristem tissue are able to divide and produce cells that once again lost the ability to divide but mature to perform specific function, and we call it as redifferentiation. Now, what is redifferentiation? I told you meristematic tissues are going to transform into permanent tissue, and we call it as dedifferentiation. Okay. This phenomena is going to, you can just check this flow chart. This is absolutely uh, easy to understand, and you can just follow this. Okay. So what is redifferentiation? Meristematic tissue are able to transform into permanent tissue back again. This is known as dedifferentiation. So let's some of the tissues in a woody dicotyledonous plant that are the products of redifferentiation. All right. So we can say that the parenchyma cells that are made to divide under controlled laboratory condition during plant tissue culture. So what would you call parenchyma tissue transforming into in the lab into specific tissues? Then we can call it as redifferentiation at that point of time. Okay. So now let's recall the section 13.1.1 over here, and then we will understand what is happening over here. Okay. So let's understand. So development, sorry. So we need to understand that we have mentioned that the growth in plant is open. Okay. This is important. Why growth is plant is open? Because the cell division is happening continuously. Okay. The cell is dividing at any point of time. You will see that the cells are dividing, and it can be intermediate or indeterminate or determinant. Okay, so they will keep on dividing and dividing. So that's why we are calling it as growth in plant is open. We may say that even differentiation in plant is open. Okay, so differentiation in plant can also be said open because cells and the tissues are rising out of the, so I'll just underline here. So please mark it in your textbook as well, that this the differentiation is open, okay, in plants, because the cell and the tissue arising from the same meristem have different structures of maturity. Okay, so the final structure at maturity of a cell tissue is also determined by location of the cell within. We need to look at the location as well. For example, cell positioned away from the root apical meristem differentiate as root cap cell, while those pushed to the periphery matures as epidermis. So location also matter, and what it will get transformed into, that depends on the location. Okay. So this is what we have understood about differentiation, redifferentiation, and dedifferentiation. All right. So let's understand this para as well. So what is development? So development is a term that includes all changes. Okay. It will include all changes an organism goes through during the life cycle. Okay. From germination of seed to the census. So this is basically your development. So there is a diagrammatic sequence of process which constitute the development of cell of a higher plants in a given figure. It is also applicable to tissues and organ. So development is starting from the germination till it reaches the census. Okay, let's have a look at this diagram. So this is a plant. This is a flow chart of a plant growth. So plant follows different pathway in response to environment or phases of life to form different kinds of structure. So plant also respond to the environment. Depending on the environment, they are going to make certain structures. Okay. So before that, we need to understand this diagram as well. What does the diagram says over here is, you can see meristematic cell. Meristematic cells will undergo differentiation, where you will find that the cell is forming a mature cell. Maturation, elongation, and differentiation, plasmatic growth, all these things are going to happen. Cell division is going to happen and lead to the formation of a mature cell. This mature cell will also undergo senescence, and after certain time period, you will see that it will undergo death as well. So this is the sequence of developmental process in plant. So can you see how the developmental process have started off from here, which is meristematic tissue, and ending up at the senses that could be your permanent tissue. Okay. Now there is an important point over here, plasticity, which is very, very important. Let me take some examples of plasticity over here. Is sometimes plant also uh, show response to the environment, and they are going to form different structures. This is known as plasticity. Now, let's take an example of a heterophyly in cotton or coriander or larkspur. So you can see over here, heterophyly in A is your larkspur and B is your buttercup. So during the juvenile phase, you can see that it has transformed the structure like this. But when it is adult, the structure looks like this. This we are talking about plasticity depending on the requirement or depending on the environment they are going to form the structure. In such plants, the leaves of the juvenile plants are different in shape from those in mature plants. Okay. So on the other hand, difference in shape of leaves produced in air and those produced in water in the buttercup also differs. This is known as heterophyly. Okay. Heterophilous development due to environment. Okay. So this phenomena of heterophyly is an example. This example of heterophyly is known as plasticity. So you can see over here how in the buttercup, if it is if it is in terrestrial part, then the leaves will look like this. If it is moving into the water, the water habitat means the structure of the leaf will look like this. So this is basically your heterophyly, or you can say this is a phenomena of heterophyly is an example of plasticity. Now let's understand. Thus, growth, differentiation, and development are very closely related event. They are absolutely related. Growth, differentiation, and development. Okay. So broadly, development is considered as the sum of growth and differentiation. So we can just write over here, development is equals to growth plus differentiation. Okay. Growth and differentiation. That is basically your development. You can write down this somewhere. So growth, development in plant, both growth and differentiation. Both growth and differentiation is responsible under intrinsic and extrinsic factor. So this we will be studying, and we have to understand what are intrinsic is internal factors or intracellular genetic factors, you can say. Okay. And extracellular could be the other factors which are external, like light, temperature, water, oxygen, nutrients, and these. Okay. An internal factor will include chemicals such as growth regulators, which we will be studying in the next session. I hope this is absolutely clear to you all, like what is plasticity and what is heterophyly and what is development. And development includes maturation and cell division. All right. So we have discussed about differentiation and re-differentiation. All right. Thank you so much.